In order to do these metabolic processes, we need enzymes for a lot of these steps. Enzymes are very specific. They must fit together with the molecules they are acting upon in a lock and key fashion. Enzymes are called catalysts. They catalyze reactions by decreasing the activation energy needed in order for the reaction to proceed. In essence, the enzyme speeds up the reaction. Now, without the enzyme being present, the chemical reaction will still take place. However, in order for the human body to digest one hamburger, it could take decades without the help of enzymes. So even though the process would proceed without the addition of the enzyme, having the enzyme present allows our body to metabolize things fast enough so that we can continue to live. Otherwise, we would starve to death. Enzymes are not considered reactants or products in chemical reactions. They are not consumed and they are not altered when they catalyze a reaction. So for this reason, they are reusable. There are different factors that are going to affect enzymes. Only a couple of them are shown here. So there's a narrow range of temperature that enzymes will be able to function at their best. Also, if there's a shape change due to pH or temperature or maybe the binding of some type of inhibitor or substrate can also be a factor here. So let's look at two different types of competition that we see for these enzymes. We've got competitive inhibitors and non-competitive inhibitors. A competitive inhibitor is going to compete for the same binding site that the substrate would like to use. We call this the active site. Whereas in non-competitive inhibition, we see that the inhibitor is actually going to bind to a site other than the active site. So they are not competing for the same site. However, when this inhibitor does bind to what we call this allosteric binding site, we actually see a conformational shape change in the active site or the substrate binding site. Remember, it has to fit in the lock and key mechanism. So when there is a structural shape conformational change with the substrate binding site, this is not going to allow, this is going to inhibit that substrate from binding. So in this way, it's going to allosterically inhibit as a non-competitive inhibitor of the enzyme.